Welcome back, friends. In this video tutorial we'll be talking about Helicobacter pylori and the infections caused by Helicobacter pylori and the treatment of that infection in a little bit amount. So let's begin with this. First thing I want to talk about the disease that are caused by Helicobacter pylori, the signs and symptom of the disease. Now in the major scale, Helicobacter pylori cause diseases like gastric ulcer. Usually, most of the time the disease remains asymptomatic in nature because people always often confuse this kind of symptoms with milder gastric upset and indigestion problems and several things like that. That's why it remains kind of asymptomatic, but it can cause severe inflammation or damage to the stomach lining. In certain cases, when it starts damaging the stomach lining, damaging the mucus layer of the stomach, and as a result of that the acid content of the stomach start to hamper or degrade the cells of the stomach lining. As a result of that, it can result in stomach ache, abdominal pain, acid reflux, nausea and vomiting. So these are all symptoms of Helicobacter pylori infections. Now if it is untreated for longer time period, Helicobacter pylori infections may cause severe complications like gastroesophageal reflux disease or GERD or GERD. Now in this case what happens, the gastric content of stomach kind of effluxed out through our esophageal wall, and as it is made up with acid most, that's why this acidic content of stomach juice can cause severe damage to our esophageal wall. And it can also cause peptic ulcer. In the final stage it can cause peptic ulcers, normally duodenal or gastric ulcers, and in very few cases it may cause cancers of the esophagus as well as cancer of the stomach, but it's very rare, but it may cause it. Now let's talk about the genome of Helicobacter pylori. It consists of a large diversity of strains actually, and among many variety of strains, only 3 of those strains have properly sequenced genome, but still we don't know the function of all the genes of that sequenced genome. But we know that that among those genome, 40 kilobase per long section of pathogenicity island, it is called CAG pathogenicity island, which is very important for the infectivity caused by Helicobacter strains, Helicobacter pylori strains. Okay, now normally the Helicobacter pylori strains obtained from humans who are carriers of Helicobacter pylori, we generally won't find that CAG pathogenicity island there, but for if we collect it from the infective property or infected people, then we can find the CAG pathogenicity island playing an important role there. Okay. And now what and normally the lower GC content of this CAG pathogenicity island of the Helicobacter pylori genome is showing us that normally what we know, if we are having low GC content, high AT content, it's a normal indication that they may get this kind of genes and gene sequence via the horizontal gene transfer processes. So it is a cause of the horizontal gene process. And also we can find that the genome contains significant number of transposable genetic elements, and they are also present in different sections, and and as a result of having many more variety of transposable genetic elements as well as those elements for pathogenicity island section, we may conclude that they are getting this infective property from other bacteria via horizontal gene transfer, and this transposable genetic elements are helping them to transfer the content of a region which is responsible for disease causing section from one to another. Now in this in this picture we'll be talking about the Helicobacter pylori pathogenesis and the overview of this process. Now there are 4 different processes sections. First is the adhesion of the Helicobacter pylori to the host cell. Second is minimizing the acid content of the stomach. That's the most important and unique point about the Helicobacter pylori infection. Third one is the colonization of the Helicobacter pylori, and the fourth thing is the degradation of the epithelial cell of the stomach lining by the Helicobacter pylori. So if we begin with the first case, the attachment of the Helicobacter pylori to the surface epithelial cell, and in this case what we can see, the bacteria kinds of propel itself using its flagella. It migrates through the mucus layer and through the gastric fluids and mucus layer. Finally it reaches the epithelial cell and it adheres to the epithelial cell using a small extension of cytoplasm called fimbriae. So using fimbriae they are kind of interacting with the epithelial cells of stomach, and also using another molecule called adhesion, which is helping this Helicobacter pylori to attach with this epithelial cells. Very important to have that interaction. Now adhesion is the first thing that must occur prior to the engulfment of the bacteria inside. Now once after adhesion is completed, this bacteria wants to minimize the acidity of the stomach content, because we know inside the stomach there are acid secreting cells, and those cells are secreting acids outside. Now the stomach cells now this bacteria this uh, Helicobacter pylori must neutralize it, and they neutralize it by secreting some alkylating agents like ammonia, right? So in this case we can see they secretes enzyme called urease. Using urease, they can break down urea into ammonia and carbon dioxide. Using both ammonia and carbon dioxide, they can reduce the acidity of stomach juice, and as a result of neutralization of acidity, the bacteria can live there for longer period of time. Now after neutralizing the environment, what they can create virtually a kind of micro environment. As you can see here, this is kind of changed in different color. So this is a small micro environment produced by the Helicobacter pylori by secreting urease, is very important. Once this micro environment is created, then more and more pylori can come and start to colonize in this particular area. And after a successful colonization, what they can do, they can start to secrete certain chemical mediators, some protein factors, some enzymes like proteases, lipases, and some other factors that are going to degrade the host cell epithelial, right? And after that they start to produce some other factors also, with which they can create an inflammatory response inside our stomach lining, and finally eventually it will cause the mucosal cell death in this stomach lining, right? And this is very important because the stomach is fully covered with the mucosa. Until and unless they degrade this mucosal epithelial cell, the mucosal cell lining, it won't reach the terminal cells of stomach. So that's very important. Now once after reaching the stomach, colonizing onto the stomach, the final point of this cell is to secrete certain chemical factors, certain protein factors, enzymes that are helping this bacteria to cause damage to the epithelial cell, right? And what are those factors? Now in this picture we are going to see 7 such factors that are causing damage to our host cell. So let's begin. The first thing is the flagella. Using the flagella, this bacteria can usually migrate and move through the stomach. It can migrate through the stomach and finally reach the stomach. That's very important. And second thing is urease. We know this enzyme helps to neutralize the gastric acid by converting urea into ammonia and carbon dioxide. Now the third important thing is lipopolysaccharide. It helps in the adherence of the host cells, and also it causes the inflammatory response, because this lipopolysaccharide can act as endotoxin. We know it is an LPS layer. It can act as endotoxin to produce a toxic shock syndrome and toxic shock conditions in the host body. Fourth thing is the outer membrane proteins. For example, here these proteins designated here with this orange color sections, these are the outer proteins. Now these proteins are important, and it is required to adherence of the bacteria to the host cell membrane. The fifth thing is the exotoxin. Now this bacteria is not that much dependent on the exotoxin for the infectivity, but it may produce exotoxin called the vacuolating toxin. It's the example VAC A or vacuolating toxin A. Secretion of this toxin to the epithelial cells start to degrade the cell, and it eventually degrades the cell by forming vacuoles inside the cell. Now the sixth thing here is the secretory enzymes. Enzymes like mucinase, protease, lipase. Now protease can degrade protein, lipases can degrade lipids, and mucinase can degrade mucous lining or mucus layer of the uh, stomach. And seventh thing is the type 4 secretion system, because we are going to see in this Helicobacter pylori they deliver all those important components inside the host cell via the type 4 injection system, right? It's kind of syringe like system, as you can see here. It's a kind of cartoon picture, but still it's kind of syringe, molecular syringe, destined to deliver certain important factors inside the host cell. It's kind of pili like ejection. Okay. And finally, seventh, the effector molecules that are required here are actin remodeling complexes, because they need to remodel actin prior to stop the fusion of lysosome with the endosome. And also they need to recruit certain factors which will induce interleukin 8, which eventually cause the inflammatory response, as well as they need to produce some apoptosis inhibition factor. And all of these factors are made using that CAG-A, which is a part of CAG pathogenicity island. Okay, now let's talk about major sections of the pathogenicity. uh, First is adhesion. Now Helicobacter pylori is also very important, and for the adhesion of the Helicobacter pylori, it needs to have carbohydrate onto the surface of the host cell membrane. So it must have the carbohydrates or lipids, but it usually interacts with this carbohydrates, host cell carbohydrates and lipids, using certain adhesin molecules. For example, one such adhesin molecule is BAB-A or BABA. Using this BAB-A molecule, it can interact with Lewis B antigen displayed by the surface of stomach epithelial cells. So it's a typical example of the adhesion of Helicobacter pylori with the epithelial cell. Then the inflammation. For the inflammation to occur, they require this type 4 secretion system. Using type 4 secretion system, this Helicobacter pylori can secretes its own peptidoglycan segment of its own cell wall. So it can cut its own peptidoglycan section from its own cell wall and secrete this or inject this peptidoglycan inside this host tissue or host cell, so that host cells start to develop start to express certain factors against it, factors like interleukin 8 against it. Now the injected peptidoglycan can be recognized by the cytoplasmic pattern recognition receptor or immune sensors. For example, here such immune sensor is NOD1. After recognizing this section by NOD1, they start to secrete certain factors like interleukin 8, which call upon many more immune cells like macrophages, like neutrophils and other important lymphocytes molecules, so that the inflammation is resolved. Okay, so you can see here the result of inflammation, that secreting all those factors are important to bring upon the IL-8 and other factors to finally cause it. Now the type 4 secretion system also injects the CAG pathogenesis island protein CAG-A into the stomach's epithelial cell, which disrupts the cytoskeleton of this stomach epithelial cell. So everything is important. Remember I've talked about the cytoskeleton remodeling molecules, apoptosis inhibitory molecules. It may cause cancer if it is triggering that. So many things can occur in this process. Now this CAG-A protein can also regulate the host cell gene transcription. So it's a kind of controller of this process. You can see here the damaging conditions that are caused by Helicobacter pylori in the very terminal stage of the infection. The gastric ulcer, it's a very basic stage, then it can form the gastric tumors. That is very, very dangerous. It's kind of a harsh condition. They are forming cancerous. They're going cancerous. Now other toxic factors produced by Helicobacter pylori are, besides ammonia, there are other factors like vacuolating cytotoxin A or VAC-A. We have talked about it. As well as they can produce phospholipases, which can damage those cells and usually the cell membranes. Okay. Now there are two related mechanisms by which Helicobacter pylori can promote cancer under certain circumstances, but still it's under prior investigation. One of the machinery, it involves the enhanced production of free radicals near the Helicobacter pylori. As they start to produce these free radicals, the increased rate of cell mutation start to occur. As a result, those cells may turn into cancerous cells. It's a simple way of transformation. Now the second possible way is called the perigenetic pathway. Now it involves the enhancement of the transformed host cell phenotype by means of alteration in the cell proteins such as the adhesion proteins. So change in those protein contents is called the perigenetic pathway. It's because it depends on the gene, but directly not gene, on proteins or protein modifications. Now, the most important and interesting part about the Helicobacter pylori topic is how pylori survives the stomach acidity. Now, we know that they produces ureases, but exactly how? Now, the machinery is very basic, that they produces urease, which is an enzyme to break down urea into ammonia and carbon dioxide. So they are breaking urea into ammonia and carbon dioxide. Now, the ammonia is then converted into ammonium by taking a proton, because there are a lot of water out there, so they can usually take a proton from there, because acids are there, HCl is there, so protons are plenty of protons are there. So the ammonia will be converted into ammonium, and the carbon dioxide that resides there, that carbon dioxide will interact with hydroxyl ions that are also pretty common there, because water is always available. So the carbon dioxide will interact with hydroxyl to produce bicarbonate, and this is the bicarbonate which neutralizes the gastric acid. It's not actually ammonia, because ammonia is converted into ammonium and then it's done, but the carbon dioxide binding with the hydroxyls from the surrounding is finally converted into bicarbonate that reduces the acidity of that environment, and the reduction of acidity is very vital. So this is a way of how it's done, this is the way that we have understood, but behind this process there are many genes are working, many things are working, still all of the machineries are not clear yet, research is going on. Now if you look at the relation of Helicobacter pylori with different pH, it's very important, because the life cycle of Helicobacter pylori and how the pylori work and how the pylori live, it depends on the pH actually. So if we look at three different section of pH, one is pH 5, kind of moderate, then pH less than 4, very acidic, and pH 7 is kind of neutral. If we look at the pathogenesis of pylori in different pH, like first if we look at it at pH 5, it produces more complex O chain forming Lewis antigen and terminated by different unit, and then it mimics the human gastric mucosal cell surface glycoconjugates, and may cause invisibility of Helicobacter pylori in most of the host in normal pH 5. Finally, then it induces the reactive T cells to initiate autoimmune diseases and may cause autoimmunity in these conditions. Similar thing happens if the pH is kind of 7, because again it produces Lewis antigen, and anti-Lewis antibodies may be introduced that binds with the gastric mucosa and can cause damage. So again autoimmune response by self immune system against self cells, so it's a kind of autoimmune in this case. So in pH 5 as well as pH 7 may result in autoimmune system. But if we look at pylori development at less than pH 4, it produces urease. Now if it is having a pH of more than pH 5, it generally does not produce urease, but if it is a pH living in the pH less than 4, it produces urease, metabolizes urea, neutralizes the gastric acid present there, produces mucosal lesion, and finally may cause autoimmunity. So finally everything is kind of leading to autoimmunity in this case. Okay, now what can be the diagnosis for that? Now diagnosis for Helicobacter pylori can be blood tests, it can be it can be urea breath test, and sometimes sputum not sputum test, stool test, but in very few cases. Okay, now normally blood tests, urea tests, and stool antigen tests are being conducted. So if you look at the blood tests, we look for the presence of antibodies against this Helicobacter pylori. You need to find those antibodies. Now, the one important factor about these blood antibodies that they can persist for years after the complete irradiation of Helicobacter pylori with antibiotics. So they are good to determine if the antibiotics have successfully irradiated the they're good for determining the infection, for diagnosing the infection, but they are not at all good for determining whether the antibiotic worked properly or not, because even if the antibiotic works properly and the infection is eradicated, still you are going to find a lot of antibody in your blood after the several years of completion of the of this therapy. Okay. Now the second process is called the urea breath test, and this is very important. This is safe, this is easy, and it's kind of accurate test for the presence of Helicobacter pylori in the stomach. Now how we can determine it? For this to occur, the patient need to take urea. What is the foundation of the experiment? That we know that pylori produces urease, it breaks down urea into carbon dioxide and ammonia. So it's the idea that if the patient take the tablet with radio labeled carbon as urea, so C14 urea is taken up by the patient, and then in stomach the urea is broken down into carbon dioxide. Now if this urea is attached with the C14, which is the radio labeled, then the carbon dioxide they are going to produce is also radio labeled with C14. So it's convention that after taking the urea, that patient will produce a radio labeled carbon dioxide in their breath. So if we measure the radio label condition using scintillation counter, we can find we can tell that yes, that patient is infected with this Helicobacter pylori. Then we can say that the test is positive. Okay. And if the test becomes negative shortly after irradiation of the bacterium from the stomach with antibodies, we can also find that the test shows negative. So it is very good that if this test says positive, we can say yes, infection is going on. If it's negative, no infection. Is very basic. Now another test like endoscopy can be done, but it's kind of painful, but we can rely on endoscopy. So it's a kind of flexible tube that is inserted inside, having a camera attached to the end terminal. It will see all these things, what is going on inside. You can see, you can visualize it actually, and then we can take a scrap of the tissue from it. You can look for after staining whether Helicobacter pylori is there or not. And we can also find the stool antibody test. This is kind of most recently developed test for the detection of Helicobacter pylori. Now in this test, what we can do that we can diagnose with sample of stool, and what we do here, we add some antibodies which will bind with the Helicobacter pylori if the pylori present there with in the stool. And like the urease breath test, in addition to for the diagnosis properly, we must rely on more than one test, not only one. Even if we know that this urea breath test is kind of good, but still we need to rely on more than one. For example, urea breath test along with that stool antibody test can be done. Then finally, the treatment of Helicobacter pylori and pyloris. In this case, they're difficult to eradicate from the stomach. Now, why they are difficult to eradicate from the stomach? Because it is capable of developing resistance to commonly used antibiotics very fairly. Now, the antibiotics that we usually use via the horizontal gene transfer, they are much more resistant. So not only one antibiotic can prevent them, so we need to use a combination therapy along with a proton pump inhibitor. That's also important. The proton pump inhibitors, one such example is omeprazole. So we need to take a proton pump inhibitor for sure. Along with that, we can use a combination of antibiotics like amoxicillin, clarithromycin, or with proton pump inhibitor we can take metronidazole, tetracycline, and bismuth subsalicylate. Okay, so these are the prescribed sessions of Helicobacter pylori treatment, but you need to be very careful because you need to check the food you take. You need to look after your digestion, the amount of food you're eating, because many things are hampered if the Helicobacter pylori infection goes too far. But if we detect it in the very earliest stage, it can be eradicated. It's very easy. Okay, so that's kind of it, and I hope it's helpful. Thank you.